5G in healthcare, the need of the hour. Key takeaways: treatment on the go, chronic with drive, prioritizing patients, better hospital management, real-time analysis of patient during oncology treatment.
Thank you, Maya, for those insights. Uh, I would like to request everyone that you could follow us on Facebook, LinkedIn, and Twitter on the IT India handle, and you could use the hashtags FTC and Decode the Future. One more exciting announcement is that before we close the ceremonies for today, we will have a Q&A round with Sir Robin Saxby, exclusively with you guys with here today. So please stay back, and while we finish off with the healthcare, we have a short discussion on mobility. Post that, we will have Sir Robin Saxby for a Q&A session. So please stick around.
Thanks, Suman. I now invite Dr. Kiran Aatre to come on stage and enlighten us about the intersection of advanced technologies and life sciences. We will hear from a company that is at the cutting edge of electronics, digital technology, and display systems, but it is seemingly ancillary to healthcare technology. How does a semiconductor equipment manufacturing company connect to the medical tech, and how do they see their role in the future of personalized medicine and cancer diagnostics? Welcome, Dr. Kiran. And to introduce Dr. Kiran Aatre, he's the Deputy Director Business Development at Applied Materials in Bangalore, has close to 15 years experience in developing medical devices and associated technologies, has been responsible for business and technology development in new and adjacent area with additional responsibility of ecosystem and startup outreach. Mechanical engineer from Bangalore University by training, received his MS in engineering science from Penn State and PhD from Arkansas in microelectronics and photonics. Started his career in research, was a part of Bosch in his journey, and now he's developing or looking at development of low-cost point-to-care non-invasive medical devices. That's what he did in Bosch. He's an adjunct faculty at the Manipal Academy of Higher Education, and now with Applied Materials working on the cutting edge of technologies. Welcome, over to you, Kiran, and welcome for this.
Good morning, everyone. I guess I'm standing between all of you and lunch, so I'll try to keep it as interesting as possible. So what I want to talk to you today is about the semiconductor equipment world. You heard a little bit about it from Suraj yesterday. I will recap some of what he said and also try to give you an idea of what we are doing in life sciences. A lot of it is thought experiments. How can we as technology developers try and stitch this entire ecosystem together? Can we provide technology solutions that could help the future of life sciences? We saw this slide yesterday, so I won't spend too much time on it. We are a 55-year-old company. We are based out of the Bay Area in Santa Clara, to be specific. We have, this slide is slightly dated, as Suraj pointed out. We're at about 7,200 employees right now with our headquarters in Bangalore. We have centers all across India. Our research lab is at IIT Mumbai. Most of our people work out of the Bangalore office here in ITPL. So we look at a lot of design aspects, a lot of AI, a lot of software, a lot of services that we provide for our existing customers. We heard some of this yesterday as well. Our primary business is to miniaturize electronics. Can we push electronics into the as smaller and as tightly packaged a system as possible to enable all the technologies that you use? We invest about 1 billion annually in R&D, and this is primarily in the semiconductor space, of course, which is our bread and butter. But we also use a lot of that to look at technology adjacencies through our officer CTO. So the officer CTO is tasked with trying to take what we know and try and identify what else can we do with it. Now, whether we run this as a business on our own, whether we spin this out, or whether we license it out to somebody else, all comes down to what kind of business models we like to follow. But why life sciences? Why is it that we think we can do something in life sciences? So I think one of the important aspects is to understand how does applied even connect with life sciences. What we do is not creating new materials. We manipulate the materials. We create features that can be used in various materials and try and etch material and remove material at the nanoscale. Now, when you're able to do that at the nanoscale, we also need to be able to measure that and assure the quality. As Suraj mentioned yesterday, if we cannot do this repeatably for every single device, you're going to have variation in performance across spectrum. Can I pick up that defect within those 10 nanometer features that we have put down? And can we actually tell people this is where we are? The analogy that we like to give is that this is the equivalent of trying to image a coin which is lying on the street while flying overhead at 2,000 miles per hour at 5,000 feet. That's a scale at which we image. And you should be able to read that lettering on the dime, read the D, and make sure you're able to identify every single feature of that. This is the scale at which we operate. So we think this is kind of similar to what we do in life sciences. If I can image something at 15 angstroms, very similar in size to RNA and DNA, can I pick that ability to image at high speed or high resolution and try and translate that to life sciences? That was the idea behind which we started looking at life sciences. And some of the potential areas that we look at for our engineering expertise spans right from clinical diagnostics all the way to drug discovery. So we are trying to look at how do we map out this entire space. What can we create in this environment? I'll talk to you briefly about some of those, hopefully without upsetting our IP lawyers. But this is where we are. The first thing I would like to point out is a product that we have created called Mimamsa AI. This is a product for use in breast cancer diagnostics right now. We plan to expand the portfolio a lot more. It's an AI tool which enables pathologists. Now, there are several players out there as well who do the similar thing. We do believe we have some advantages in terms of the way we've built the algorithm and the way we want to stitch all these features of the product together. We do have a team available at the FTC right now. We have a stall run by Applied Materials, which is in the exhibition area. I do encourage all of you to go down there. The team would be happy to walk you through what they've done, how they've built the entire system, and what is it that they see are the salient features of this device. But moving on to this, in March of last year, we spun out a company called Veranome. Veranome Biosciences. You can look up this information. It's available on LinkedIn and a few other sources as well. This was kind of the jump-off point for our forays in life sciences. What we wanted to do was look at spatial genomics. Can I start identifying thousands of markers in fresh frozen tissue? There are several advantages of using fresh frozen tissue versus using formalin-fixed tissues right now. It retains the DNA and the RNA in their native state, giving you a much better understanding of what actually is going on in the system. Can I take this image? Can I overlay it with these thousands of markers that we're able to identify? Can we take some of that high-powered compute and the high-resolution imaging, and can we start getting a lot more information out of the cell? This is what is loosely classified as spatial transcriptomics. There are competitors in this space as well, but what we are doing with the imaging is significantly different from what the other players do. And we'd love to talk to you about this as well at some point of time. But what do we do in this? How do these things, various pieces sit together? We want to understand, can we look at spatial RNA? Can we do multi-omics analysis? Can we tie this in with traditional histopathology studies? Can I take the knowledge that we have from our Mimamsa tool and from our Pisces tool, overlay these two, and create a much larger repository of information which can be used across the board, right from research to pharma to clinical R&D as well. So this is the crux of what we want to do in this space in pathology and the intersection with genomics. AI, of course, is going to play a tremendous amount of things, but as our gentleman from HCG pointed out a little earlier, this is not just GBs of data, this is terabytes of data. Computing this, running through this is a difficult job. How can we stitch all this in with transmission of data? Can we bring all this on the cloud where researchers can access it from everywhere? Is there some way we can bring all of this in place and get the kind of information that we are looking for? What is that information? Can we use this to infer the kind of cell subtypes that we see? Can we use algorithms and the associated gene expression along with morphology, which you get from traditional pathology studies? Can I build this entire gene expression database and try and see what is it that is actually happening within the body? To put it very crudely, when the mRNA exits the nucleus, did it make a right? Did it make a left? Which cell did it take the information to? And because of that information that it took to a cell that is either the correct cell or the wrong cell, did it trigger the development of cancer? This is the kind of information, if we think we can start building a database of this kind of information, we can actually provide this to pharma companies, which will help them tailor their medication to understand, okay, I can now target this specific kind of mRNA which has sent the wrong message to the wrong cell and has triggered cancer in the person at some point of time. Can this be a feeder to some technology like CRISPR? Can we start clipping the cells and say, okay, even though you've sent the information in the wrong place, can we take some remedial action to make sure this doesn't trigger cancer in somebody else? That is where we think this entire system will start falling together. Now, we as technology developers can do all of the imaging, all of the analysis, but we still require the ecosystem that does the rest of the work. To give you an idea what kind of complexity the system has right now. Traditional spatial transcriptomics, you need to start dicing up the cell into smaller sections. You use microarrays, and every single one has to be manually examined. With digitalization, what I can do is image this entire tissue at various points of time. I can start picking up what the status is. We can start looking at what are the poor prognostic markers that we have had, and we can map out this entire system to tell you without spending hours and hours of time or even days and months in trying to analyze what's happening in those fixed tissues. This is the dream that we want to chase after. And what else can we do? Can we look at advanced imaging systems? There's a lot of literature out there about use of hyperspectral imaging in agriculture, in defense applications. A tremendously powerful imaging tool. Can I take this hyperspectral imaging tool and train it onto existing histopathology and genomic studies? Can we pick up those spectral differences at the molecular level and give you a much better understanding and overlay that with your information that you've already got from genomics and pathology? This is what we think will happen in the future. Who's going to develop this? The jury's still out. We can see where this goes. This is just a start. We also try to look at what we can do as an equipment manufacturer. Can we create equipment which will examine every single tablet that goes through as an inspection tool? This is something we've been doing for some time, and we do have some customers lined up. This is actual equipment that we use. This is easy to do for us. Inspection tools, we know how to work with this perfectly. You want to do the industrial scale? Got that solved. We can have that resolved as well. What more can we do beyond that? So applied, as I mentioned earlier, we do a lot of atomic layer deposition. Can I take the power of that atomic layer deposition and give it to pharma and say, now I can start coating every single particle of drug? Why is that useful? You now have a much more uniform coating of the drug. As you can see in the second point over here, proof points, uncoated APIs and coated APIs, they behave very differently. The way the drug breaks down, the way the uniformity of the molecule changes as you start depositing the atomic layer uniformly, it helps. Now, can this be used to trigger when the drug releases? Can we have a tailored drug which will release at various points of time? Can I create a system by which the initial molecules are released in the first 10 minutes of ingestion? Can it be released after 12 hours, after 18 hours? Will this help in treatment? This is something we will need to study. And of course, can I actually have this molecule go to a specific site? We all know cancer areas will be inflamed. Can I take this drug, send it to a specific place, have it released in controlled quantities, and create much better treatment regimes without affecting the entire body? This is what we want to do with our coating capabilities. So what we can do, uniform conformal coatings at nanometer level precision, regardless of particle shape or size. We can solve all the coating problems, but what can we do beyond the coating is what we've got to figure out with the pharma companies as well. And it doesn't just stop there. What can a semiconductor company do? We all know of nanopores, what the issues with DNA sequencing are, how long it takes. Can we create a faster, cheaper, much better way to sequence DNA? Material is material as far as we are concerned. Can we etch away the material and create a nanoporous membrane which is a lot more uniform? This is again a thought experiment that we tried with a few startups that Applied has invested in, and we'd like to understand what is that we can do. So we have created a high volume processing wafer on silicon. Can we create nanoporous silicon which is extremely uniform, can reduce your signal to noise ratio, and you get a better understanding of exactly what the DNA is providing in the system. So these are some of the thoughts that we had on where we do see our role in terms of the the future of technology in life sciences. We also do have a smart factory which we are trying to set up for the pharma industry and for manufacturing industry. Of course, that's what we specialize in. But can we take all of these various bits and pieces, stitch them together, create the continuum that we had alluded to as Applied's role in the entire life sciences space? This is what we do right now. This is our CTO playbook. What we like to say, think big, fail fast, test small, learn quick. We would love to do this with multiple players. We work across the board with academia, with various industries. We engage with customers at a much earlier stage. We do not want to spend millions of dollars developing something where the customer says we don't want it. Can we create these test beds where everyone is satisfied with where we are? And the final thought that I would like to leave all of you, this is something that the host of the previous session alluded to as well. This does take an ecosystem. Cancer is not a disease, it's a condition. It's your own body fighting against you. It's evolving constantly. Is there something that we can learn from this? And I think the biggest takeaway that we should all have from COVID is if we don't pull together, nature's going to find the way to beat us and beat us badly. We've got to pull the various ecosystem pieces together, whether it is a technology developer, a device developer, pharma, life sciences, academia. If we do crack this together, this is something we can solve. And we are constantly on the lookout for partners that can bring various pieces of this larger jigsaw puzzle. So if you would like to talk to us, we encourage it, we welcome it. Please do feel free to reach out to us. This is the Applied Materials hashtags that you can try and reach out to us. And of course, if you would like to talk to us specifically, this is my coordinates. You can pick up my business cards from the stall that we have, and we'd love to hear from you. Thank you.
Thank you, Kiran, for that scintillating speech. To me, it looked like a fiction story. And really, if we were to sort of fast forward our lives and imagine what this capability can do to us, it is mind-boggling. And more than that, I'm really heartened to see non-traditional companies like Applied Materials who are in semicon coming into and challenging the domain of healthcare. And that's really, really interesting. So thank you so much for educating us on what you guys are doing, Kiran. Can I request Professor Srivastava to come on the stage and honor you, please?